Hello, this is the Knowledge Catalog, and this is another topic on biological science. As you notice, we are not going to feature any learning competency from junior high school, but if you may want to use this video as an advanced lesson, perhaps for your senior high school or for your college level instruction, you may, of course, do it. Also, if you want to have a copy of this presentation, you may contact me through my page, Knowledge Catalog. You can actually go to it through the link facebook.com/knowledge catalog / Let's begin. Some environmental chemicals can be genotoxic. They can also be non-genotoxic. So when we say genotoxic, they can harm the genetic material, which is the deoxyribonucleic acid or DNA. When it's non-genotoxic, then it means it's otherwise. So yeah, they don't harm the genetic material. Some chemicals naman, they may have no carcinogenic potentials on their own, but they may greatly enhance tumor development when exposed to them. This now is mediating tumor promotion. So even though the substance is non-genotoxic, it can still indirectly promote cancer by enhancing tumor development, and I hope that you read that along with me. Okay, so the connection between genes and cancer. So the first connection is proto-oncogenes and oncogenes. So what are they? Well, basically, they are genes. Cell division is regulated by several cellular proteins which are produced through instructions coming from genes. This means that genetic mutation may result to the regulation of cell division. So, ano ibig natin sabihin doon? Ang cell division, para mag-start siya or mag-take place siya, for it to happen, it requires several signals. And those signals are proteins. And these proteins are coming from specific instruction, and those instructions are coming from genes. Therefore, if there will be any, say for instance, mutation sa mga genes, it will result to deregulation of cell division. Okay, so proto-oncogenes naman are genes that stimulate normal cell cycle. Again, proto-oncogenes are genes that stimulate normal cell cycle. So yung usual na pagde-divide ng mga cells through mitosis, it is instructed by these proto-oncogenes. They are found in various levels of signal transduction for control of cell growth, cell proliferation, yung pagpaparami ng cell, and cell differentiation, or yung pagtuturn ng cells into different forms. So, yeah. However, point mutations, insertion mutations, point mutations such as insertion mutations, gene amplification, and chromosomal translocation, as well as changes in the expression of some oncoproteins can activate proto-oncogenes. Yung mga mutations na to, they can activate proto-oncogenes para maging silang oncogenes. This means that its proliferative activities are now deregulated. Ang tandaan, okay, ang proto-oncogenes, they are stimulating the normal cell division. So it means that since they mutated now, they were now activated, so to speak. Their activities now for their proliferative activities are now deregulated. So hindi na yun kumbaga out of control na. Okay, that is the point. Kasi nga naman nag-mutate na siya. Okay, now, the second connection is with tumor suppressor genes. Now, what are tumor suppressor genes? These genes cut off unregulated progressions through the cell cycle. Thus, they maintain cell growth. Again, kinakat nila yung progressions, yung unregulated na mga cell division through the cell cycle. Therefore, they can maintain the growth of the cell. Okay, so isa sa mga pinaka-common na na mga genes when it comes to when we talk about tumor suppressor genes is P53. Okay, again, P53 is a tumor suppressor gene. And based from my readings, hindi lang naman siya nag-iisang tumor suppressor gene. Ang p53, though, is unique. Okay? It regulates gene expression and controls several genes in growth regulation. Again, nire-regulate niya yung gene expression, tapos kinokontrol niya yung ibang genes, okay, in growth regulation, yung mga ibang genes na involved sa growth regulation. It encodes a protein with 53 kilodalton molecular mass. Thus, it's p53. Now, when the DNA has damage, ang ginagawa ng p53 is ini-initiate niya yung arrest nito, yung nung cell actually, sa G1 phase. And if-facilitate niya yung repair ng DNA. Ngayon, kapag hindi na pwedeng, actually, yung damage, halimbawa, is beyond repair na, ang mangyayari doon is, ang p53 mismo, ang magte-trigger ng apoptosis o yung programmed cell death. Again, apoptosis or apoptosis, okay, is programmed cell death. Ayan. So, that word right there. So, I hope that you are now developing an understanding, a basic understanding of the workings of cell division or yung mga signals specifically and yung mga controls na mayroon ang biologies natin when it comes to regulating this kind of very important process of dividing the cell. Kasi we are going to, our topic, remember, is abnormal cell growth. So, yeah, by the end of this video, hopefully, you will be able to see the story or know the story of how is that going to happen. While the protein products of p53 gene repress, so again, yung protein products na ginagawa ng p53 gene is, kumbaga, pinipigilan niya yung cell division or cell growth. Ang mga proto-oncogenes naman, ini-stimulate nila yon. Okay? Thus, when tumor suppressor genes like p53 are inactivated, it implies the development of cancer. So, two scenarios in there. So, ano yung mga pwedeng mangyari sa cell para magkaroon ng cancer? Una sa lahat, ang pagkaka-activate ng proto-oncogenes into oncogenes. And pangalawa, is yung pagkaka-deactivate naman ng mga tumor suppressor genes tulad ng p53. So I want you to look at this illustration of the cell. Okay, and then we're going to talk about the molecular basis of cancer. So the regulation of growth among cells, okay, is a response to various biochemical signals allowing them to develop, to grow, to differentiate, and to die. So let us make sense of this sentence. So again, yung pagre-regulate ng growth, okay, among cells, that regulation of growth is a response to different biochemical signals. Okay? And that response, that capability of cells to respond to these biochemical signals allows it to develop, to grow, to differentiate, and die. It basically, our cells, okay, they basically receive different instructions under different situations. Okay, and those instructions, they cover things regarding development, regarding growth, regarding differentiation, or even death. Now, this regulation, it depends on proteins that are based from the instructions of genes which can mutate, as I said kanina. These mutations accumulate over time until there will be a progeny of cells that lost most control over cell growth. And that is what you would call as cancer genesis. So, yung mga mutations, pwedeng mag-ipon-ipon yan. As it was said here, accumulate. Pwedeng may isang tiny mutation lang ngayon, and then next time it will be another mutation until they will be quite a lot. And then, once halimbawa na, okay, lagi na lang nagkaka-mutation, naipon na yung mga ngayon, it will come to a point when the progenies or yung mga resulting daughter cells, most of them, wala na silang control sa cell growth. Okay, so yung process na yun is what we call as cancer genesis. Ngayon, kapag nag-develop na yung cancer, there are theories that tend to describe them. The first theory is clonal evolution model. According to this theory, which was proposed in 1970, the expansion of a population of cancer cells from an individual single cell causes tumor heterogeneity in terms of pathology and molecular profiles because they have acquired genetic and epigenetic changes. Again, let us make sense of this sentence. According to this, clonal evolution model is a theory, or it is a model that describes how cancer progresses. So, since ang isang cell, normal na cell, ang nangyari sa kanya is nagkaroon ng mutation, tapos ngayon, it produced daughter cells that lost control of cell division. Ang nangyari niyan is ganito, yung mga nagiging daughter cells nila, what happened to them is that they are having heterogeneity or different characteristics. Okay? Nag-iiba-iba na. Halimbawa, dati, kung halimbawa is, say, a particular cell found in a particular part of the body is only going to produce cells that are intended for that particular part of the body. But now, since it is already a cancer cell, according to the clonal evolution model, what happens then is that nag-iiba yung nagiging result na cell, yung resulting cell. Okay? Because, yun nga, proliferative na masyado yung process. Hindi na siya regulated. So, nagkakaroon ng heterogeneity. Okay? Kumbaga parang nagma-mass produce na sila. So, nangyayari is yung original product hindi na talaga makuha yung original characteristics niya. So, they are having different characteristics in terms of pathology, in terms of molecular profile. Yun. The second theory naman is the hallmarks of cancer theory. So, ang hallmarks, parang parang criteria, checklist siya. Kapag nandito lahat ng mga to, ibig sabihin nun, magda-develop ang cancer. Yeah, that's actually a simpler way of describing it. So, ang mga oncogenes, so remember, these are genes na na-activate na, okay? They acquire self-sufficiency of growth signals. Remember that cells can send signals to themselves, autocrine signaling. Okay, so once a cell has acquired self-sufficient growth signals, sufficient amounts of growth signals, it can just autocrinely send that to itself so that it's going to just proliferate. Ayun. So, yeah, di ba, may mga signals talaga, na, especially for growth, for yung mga growth factors na talagang paracrine ng pagsisignal, nanggagaling sila from external sources, okay, extracellular sources. Pero, dito, okay, autocrine na to, bali, nanggagaling na yung signal sa mismong cell. I hope that makes sense. Next one is B, it becomes insensitive to growth inhibitory signals. May mga signals na nag-inhibit ng growth. Yes, that's true. And then, nagkakaroon ng insensitivity kapag nagde-develop ng cancer. Next one is na-evade na rin ang apoptosis. Remember that since inactive na ang P53, which is, the guardian of the genome, diba, P53 yung mag-i-initiate ng cell death. Diba? So, since na-inactivate na siya, or deactivate na siya, evaded na rin ang apoptosis. Next one is it acquires limitless replicate potential, which is actually also because P53 is already deactivated. And then lastly, it sustains angiogenesis. Angiogenesis is the production of blood vessels, say capillaries, arteries, and veins. Yun. Okay, so the last one is the stem cell theory of cancer. So, may mga tumors na nagko-contain ng cancer stem cells with indefinite proliferative potential, similar to adult stem cells. So, let us hold this muna, for a second, and then just, absorb what it's trying to say. It's actually correct. Merong mga cancer stem cells. And according to this theory is that these stem cells are highly proliferative. Okay, mabilis sila magparami. they're almost similar to adult stem cells. So yung mga stem cells talagang nagpaparami, okay, those kinds of stem cells. So now we know that they are proliferative. They are okay, I'm sorry you were not able to read that. Okay. We know that they are proliferative. We know that may mga na-deactivate, may mga na-activate, okay, na mga genes. So I hope that you know now why cells are abnormally growing. So there are different factors to consider. Say, yung mga sinabi nga natin kanina, kailangan yung proto-oncogenes ma-activate into oncogenes and so on. Okay, so if you want to repeat this video, it's all up to you. Okay, my explanations are right there already. So I hope that I'll see you in my next video. Okay, this is the Knowledge Catalog. It is a brand I am developing. I am a teacher creator and I am a high school student in the Philippines, and I love developing content that I find useful and needed to better my instruction. If you want to support me, you may leave a like, and then you may also subscribe to this channel. And if you want again to have a copy of the presentation, don't forget to message me to my page. Okay, the link is found in the very first slide. Thank you so much.